Okay. Well, how's it going, everyone? It's been kind of a while since I have published an article, and I want to explain why it's taken so long to get a full-length article out. Um, as you probably know, as many of you know, at least, I have been working on another project recently called Sense of Mind, which is a YouTube channel that is dedicated to spreading information about neuroscience and psychology and mental health as widely as possible, with the overarching goal of upgrading your brain so you can improve your life, really understanding how your own brain works so that you can try to better understand your own mental health issues, your own brain, the minds of others, why people act the way they do. And it is really aimed at giving a rigorous understanding of the neuroscience in a way that basically anyone with a good literacy and education can understand. So, that being said, I have been not as present with biology and life as I would have liked. However, this following article is something that I have been working on for a while, and it is really important to me. It is something that I have wanted to write for a long time, and it's something that I learned a lot about in the process of writing. And it's basically about memory and a process called neurogenesis, which we'll talk about later in the article. But I also wanted to give an announcement about some changes coming to biology and life, and part of that is going to make up for this lost time in which there haven't been many articles published. The first is something that that many people will not, that some people will not be happy about, but which I think is necessary at this point. If you're listening to this, you will have seen that this article is available partially on the Biology and Life website and only fully through Patreon. So if you're a patron, you should be able to read the whole thing. And if you are not, then you'll only be able to read the first part about memory. So I do think that that first article is a solid article on its own, and it will give a lot of information and people will learn a lot. So that is the first item. They're going to be, articles in general are going to be more exclusive, and they'll only have partial availability. Now, you've also probably noticed that this article is, this audio article is published in a few places. One is on Patreon, one is on the Biology and Life website, and the third is on YouTube. And so that is the start of a new part of the biology and life ecosystem, so to speak. There will be more audio articles and audio microblogs and book reviews that will be popping up on YouTube as well as on the website. So in the past, when I first started microblogs, the idea was that they would be available only for newsletter subscribers. And while that's largely still going to be true, what's going to happen is the ones that have been published for a number of weeks will be published on the website so that they are accessible and archived and available if people want to go back and read them, and for non-newsletter subscribers to check them out. So that is what's going to be happening in the next couple of weeks. And you will also probably notice an increased social media presence, and just in general, biology and life will be moving along faster than it has in the last month or so. So I do want to apologize for how slow things have been going, but I want to assure you that this following article is dense with research and science, and I think it's something that you'll get something out of, I hope. So without further ado, I'd like to read this article. Memory and the Making of New Neurons by Andrew Cooper-Sansone. Without memory, without meaning. The case of Henry Molaison, or HM as he was known prior to his death at the age of 82 in 2008, is one of the most famous in all of neuroscience. He suffered from debilitating epileptic seizures, which started out mild in his teenage years, but later progressed to grand mal seizures by the time he was 27. A seizure is a wave of neuronal activity that sweeps across the brain, much like the wave at a sporting event, causing widespread activity that results in the characteristic convulsions of a grand mal seizure. Because they usually result in a loss of consciousness, they aren't typically reported to be painful, but clearly they are highly disruptive to one's life and can be the cause of anxiety. Often, the causes of a seizure have to do with uninhibited brain tissue. That is, some neurons, called inhibitory neurons, are responsible for keeping other neurons from firing. However, whether due to genetic or environmental factors, these neurons can become ineffective and thereby let the activity of other, quote, excitatory neurons run wild. The brakes, as it were, have been cut. Molaison's surgeon, Dr. William Beecher Scoville, determined that his seizures originated in his medial temporal lobes, an area of the brain just behind the ears. He decided that the best course of action would be to remove portions of Molaison's medial temporal lobes so as to cut the seizure off at its source. One of the structures in that area of the brain is the hippocampus, and a large part of it was also removed. After the surgery, Molaison's seizures were partially cured, but he now faced an even bigger barrier to a normal life. He had no ability to form new explicit memories, and he lost his ability to recall some of his old memories. An explicit memory is a memory of people, experiences, events, places, spaces, or facts, as opposed to an implicit memory, the major type of which is motor or muscle memory, which results from practicing certain actions and involves different brain systems. What was post-surgery life like for Mr. Molaison? What indeed would it be like to have no ability to form new memories, a condition called interograde amnesia? For Molaison, every day, indeed every few minutes, was completely new. He met his nurse over and over again, repeatedly realized and quickly forgot that he was much older than 27, and had to be in a long-term care facility to live a functional life. He did retain his other mental faculties, as well as many memories of who he was before the surgery, so he had a sense of self that allowed him to make meaning out of his daily life, even if he was often confused. Molaison could still use his working memory, the type of memory that lasts a few minutes and allows you to, for example, carry on a coherent, if short, conversation. That may be because he still had his frontal and sensory cortices. These are thought to be critical for working memory. I bring up the case of HM because most of us take memory for granted. We simply expect that we will retain important information, or at least that we are capable of installing it in our brains with enough effort. We don't think that we could end up like HM. We often forget how, without memory, life would be meaningless, even if it would still be worth living. Time would become a slippery concept, with yesterday being as much a mystery as tomorrow. We would live completely in the present, which could be a blessing or a curse, depending on our individual temperament. Thus, memory is a biological process, and this article is about its main organ, the hippocampus, and two of its unique features. The first is the mechanism behind its crucial role in explicit memory, the trisynaptic circuit. The second is adult neurogenesis, the process of producing new neurons inside the adult brain, something that used to be thought impossible. If life is a series of events, then memory is the thread that ties them together in a continuous string, and the hippocampus is the needle to which that thread is tied. Let's examine the needle and how it pulls the thread. A memory maker. Look at the following animation of the human brain. What you're seeing in green, or indicated by the arrow in the static image, is the hippocampus, a brain region responsible for creating and recalling memories of facts, places, and events, as well as for sending them elsewhere in the brain to be stored. It's also involved in other functions, including emotions. The hippocampus is not homogeneous gray goo, but instead has a distinct pattern of organization that supports these functions, the most well understood of which is called the trisynaptic circuit. To understand the basics of how information is made into a memory, let's consider an example using visual information. Note that the following pathway is derived mainly from studies in rodents, but there is evidence that this system is comparable in primates like ourselves. The visual system. Dorsal and ventral streams. As information comes into the brain through the eyes, it travels to a relay station called the lateral geniculate nucleus of the thalamus. From there, it goes to an area of the cerebral cortex near the back of the brain, called the primary visual cortex. There, it is split into two different streams, partially differentiated based on the type of information, and then sent to different areas of the brain. One, the dorsal stream, which travels along nerve fibers closer to the top of the brain, is responsible for carrying information relating to where an object is or what it is doing. The other, the ventral stream, which travels along nerve fibers closer to the bottom of the brain, carries information about what the object is. Thus, these are called the what, ventral, and how or where, dorsal streams. However, as pointed out in a 2017 review in Nature Neuroscience, the what stream also encodes information about the spatial relations of objects, and thus the distinction between what and where/how streams is only partial. Following the further processing in their respective patches of cortex, these two streams then each go to slightly different areas of a structure called the entorhinal cortex, then onto the structure we are most interested in, the hippocampus, which will allow the brain to remember what it has seen. How the trisynaptic circuit of the hippocampus works. Here's a quick summary. We'll break down these three major steps below. 1. Information from the entorhinal cortex enters the hippocampus through the dentate gyrus, DG in the diagram below, and is relayed to an area called CA3. CA3 then sends the signal to another area called CA1. CA1 then sends it back to the entorhinal cortex, which relays it to other areas of the cerebral cortex, where it is stored as a pattern of connection among neurons. To visualize this, look at Figure 1a, adapted from Stepan, Dine, and Eder 2015, which is reference 5. See the attribution in the references section. And focus only on the red line, which shows the flow of information we just described. Now look at Figure 1b, which shows a mouse hippocampus undergoing precisely this process. The color indicates neuronal activity, with red indicating higher activity and green not as high of activity. You can see that information flows through the hippocampus like a wave. Throughout the following discussion of this mechanism, keep in mind that while it is presented as if there is a single wire going from the entorhinal cortex to DG to CA3 to CA1 back to the entorhinal cortex, the reality is more complicated. Many nerve fibers, wires, form this circuit, and each area, DG, CA3, and CA1, is composed of many neurons. In other words, there's a lot of information processing going on within this seemingly simple circuit at every step. Additionally, CA1 receives input directly from the entorhinal cortex, which provides a template that CA1 can match to the input coming from CA3. A final caveat is that it is now thought that CA2, yet another hippocampal area, aptly named due to its position between CA1 and CA3, can also receive input from the dentate gyrus and send that directly to CA3. We will largely ignore this structure, even though it may be important for understanding how memory really works, because the classical trisynaptic circuit is so well characterized and will give us a working understanding of the neural basis of memory. Step 1. Dentate gyrus to CA3. The dentate gyrus is the way station that determines whether a signal is strong enough to move through the hippocampus and become a memory. If the signal is strong enough, if the information seems significant enough that it warrants becoming a memory, it is permitted to flow from the DG to CA3. Before it does, however, the DG processes the signal to make it more distinct and easier for the rest of the hippocampus to understand. This distinction makes it so that, upon retrieval, the memory isn't confused with other similar memories. From CA3 to CA1. First, CA3 uses a densely connected set of neurons to store the pattern it received from the DG. Those neurons then interact with a comparable set of neurons in CA1, and the two sets forge stronger connections between them. This strengthening happens through long-term potentiation, LTP, where the connections among neurons become stronger by chemical and structural modifications. Step 3. From CA1 to cortex. Now that the information has been made into a distinct pattern of neuronal activity, a memory, it, or at least the label needed to recall it, can be sent back to the entorhinal cortex and eventually to the medial temporal cortices, areas of cortex on the outer surface of both the left and right halves of the brain, where it is stored and further modified upon recall. In the cortex, it seems that memories begin to form integrated networks, or webs of knowledge. To retrieve a memory shortly after it has been encoded, it seems that the hippocampus is all that is required. After a few weeks, the medial temporal cortices become involved in recall, and by the time several years have passed, the entorhinal cortex, along with the hippocampus and medial temporal cortices, are all involved in recall. How do you perceive a memory? This all sounds very tidy, but it leaves out the most important part about memory, remembering, or the experience of seeing a memory play out in your mind's eye. How does that happen? Let's see. Once again, consider the example of a visual memory. Just like all perceptions of vision, the experience of visual memories occurs in the visual cortex, an area near the back of the brain. However, before they can be experienced, memories must be retrieved. Where they are retrieved from depends on how long they have been around, as mentioned above. Long-standing memories are retrieved from a storage place in the temporal cortex, with the oldest ones requiring the entorhinal cortex to be activated. Newly formed memories, on the other hand, are retrieved directly from the hippocampus. As mentioned above, working memory does not seem to require the hippocampus. Instead, working memory is more like an ongoing perception occurring in the relevant brain regions, e.g., visual and prefrontal cortices for working memory of something you were just looking at. The process just described is for visual memories of experiences, facts, places, people, and events. Other senses have a similar mechanism, but they can differ in important ways. For example, when you smell something, the neurons responsible for carrying that signal to the brain go to a structure called the olfactory bulb, and it has a direct connection to the hippocampus and amygdala, meaning that smells don't go through nearly as many processing steps before reaching the emotional and memorial areas of the brain as do sights, sounds, and tactile sensations. In general, to experience a memory of any kind, whether it is a single type of sensory experience or a combination thereof, the brain regions responsible for that perception of that sense, for example, visual, auditory, olfactory, somatosensory, insular, or gustatory cortices, must be activated in a way similar to when you experience the original sensations. Stated another way, the conscious experience of an individual memory is a pattern of brain activity occurring in the areas of the brain where the content of that memory was first experienced. For example, you see a bird, and your experience of seeing that bird is a particular pattern of neural activity in your visual system, especially the occipital cortex at the back of the brain. Your visual system then somehow compresses or summarizes that pattern so that it can be transferred to the hippocampus, which eventually sends it to the temporal cortex, where it is stored as yet another pattern of neuronal connections. Then, when someone mentions that particular bird, the image is retrieved from your temporal cortex and then somehow activated in the visual cortex, where it is seen as an image in your mind's eye. To me, what is fascinating about all this is that memories are repeatedly transformed, edited, and compressed, yet they can always be reinflated to their original grandeur, or something like it anyway, at a moment's notice. It's somewhat like a digital file. You take a picture with your smartphone, and that image is converted into a series of ones and zeros, and you store it on your phone's memory. Not as the original image, but instead as those ones and zeros. When you go to show someone that picture, your phone retrieves those ones and zeros and then converts them back into a pattern of pixels on the screen. But our brains are not the same kind of computers as your phone or laptop, and some would say they are not computers at all. By anyone's light, however, the brain is an information processor, but the mechanisms it uses to carry out processing are quite different from those of conventional computers. This distinction is important, but it sometimes makes people think that brains are doing something other than information processing, perhaps something more poetic and less mechanistic, like pure experience or pure intelligence. But that is equivalent to saying that the brain works by magic. However, the brain is a living organ, so it is able to do things that would seem magical if one of our computers randomly started doing it. These include trivial facts, like the idea that the brain can run at full capacity on just a few bites of toast, at least for a little while. But there are more profound ways in which the brain does what would be magical for a computer, an example of which will be the focus of the rest of this article, something called adult neurogenesis. It would be the equivalent of a computer somehow building and incorporating new hardware components into its own existing structure. Okay, so that marks the end of part 1. If you're interested in hearing the rest of this, and it does double the duration, the length of the article, if you're interested in hearing the rest of it, consider becoming a patron on patreon.com/biologyandlife, or you can find the link on the Biology and Life website under membership. But also keep in mind that my policy is that if anyone ever wants to become a patron but can't afford it, to please just email me at biologyandlifeblog@gmail.com. Let me know that you'd like to become a patron, but that the pay doesn't work for you right now, and I will personally send you new exclusive articles and access to audio articles like this. So yes, just keep that in mind, but otherwise, please consider becoming a patron for $4 a month at patreon.com/biologyandlife. Thank you, and I will talk to you next time.